So hi everyone, my name is Ishan, and the presentation that I'll be doing is called Automatic Hyperparameter Tuning with Genetic Algorithms and Transfer Learning. And so the motivation behind this project is that deep learning models are becoming increasingly more deep, and uh, intricate topologies are being introduced. And so for the hyperparameter tuning, we know it can be very time intensive, and it can be difficult to do manually because the search space for the potential combination of hyperparameters is very large. And research has shown that genetic algorithms offer an alternative to finding quality solutions because it's meta heuristic. And so I want to also leverage transfer learning as a way to improve model accuracy for especially resource intensive tasks. And because transfer learning has a lot of downstream applications for clinical applications, I'll be using a pre-trained model. And so this project aims to improve model generalizability as well, because I'll be using some data augmentations to hopefully improve model performance. And so again, the goal is to use genetic algorithms on a deep learning model. And I'll be doing this on the BRATS 2020 dataset with a pre-trained CNN like ResNet. And the hyperparameters that I will be using, or the hyperparameters that I'll be trying to optimize for are the learning rate, batch size, and the number of layers. And so the main reason I found this paper interesting was because previous work has been done with neuroevolution as it relates to augmenting neural network topologies. And I read this paper as a part of my interest in, I guess, developing neural network topologies. And I thought this could be applicable also to uh, optimizing the hyperparameters of an algorithm. So the image in the top right is basically an example of a genome encoded via a genetic algorithm, and it shows how a phenotype or a specific neural network would be encoded in this example. And within a genetic algorithm, there's operations like mutation. And so the bottom image shows a sample mutation. And in this example, you're actually mutating a connection. So if you look at the top, you see that there's a connection being added from 3 to 5. So in the right sequence or in the right genome, you see a 3 to 5 connection being made. And the values on top represent the global history of connections. And so by having a global history of uh, or a global history of connections and added nodes, you're able to uh, perform crossover, as I'll describe later. And so the top one represents a connection being formed. The bottom one represents adding a node. Remember, so if we're modifying a topology and we want to add a node, this is the process you'd use. So in this example, they're adding the node 6, right? So if they want to add the node 6 where it is, the way that this algorithm works is by creating an edge from 3 to 6, and then connecting the edge from 6 to 4. And then the 3 to 4 edge is actually disabled as it's grayed out uh, in the third in the sequence. And so that's how a node is added. And so this was just a related work using genetic algorithms that I thought would be interesting to apply elsewhere. And so this is actually then used for for generation of new candidate solutions. So if you have two topologies, like the one on the left and the one on the right, and the genomes for both of them are listed above, you can actually create new topologies via uh, the NEAT algorithm that's explored in this paper by combining them via the method that you see below. And it basically takes a look at the history of ancestors for a given node, and it's able to figure out where added nodes or nodes can be either added or connections can be added. And the emphasis of this research was to show that genomes could grow in complexity as necessary, and that uh, optimizations could be made to smaller networks, and those small improvements in smaller networks actually carried forward to larger ones. And so I assume most people in this class probably don't know much about genetic algorithms, so I thought I'd give a little bit of a background about what a genetic algorithm is. So a genetic algorithm is basically a class of optimization problems, or it's a technique for a class of optimization algorithms, and it takes inspiration from natural selection in biology and survival of the fittest. So you basically start off with an initial population, and these kind of represent candidate solutions. And you evaluate the fitness of each of the individuals in the populations, and you basically rank them by some elitism strategy. It could be in order of fitness or proportional selection. There's different types of selections, but I'll discuss that later. And so in this example, the top four are the, I guess, most elite, and so those are preserved in the ranking, and the bottom four are discarded because their fitnesses aren't as optimal. And so with the preserved chromosomes, the chromosomes go or undergo crossover and mutation. Crossover acts as an exploitative technique because you're exploiting the best chromosomes in your population, and mutation exploits the uh, explorative technique because it introduces diversity into your population, and it allows you to randomly mutate genes across your chromosome to see if there's any unexplored uh, chromosomes that you haven't yet looked into. And so the methodology that I'd be using for this project is to create a binary bit encoding of the hyperparameters of a given CNN. And so the encoding would basically be a binary bit representation representing the learning rate, batch size, and number of layers. And so for a given population of size n, we'll have, or for given encoding E, we'll have n encodings representing different bit representations of the learning rate, batch size, and number of layers. And so this genetic algorithm is run for some number of runs R. And for each run, there's some number of generations G. And for each generation, you basically evaluate the fitness of each of the candidate solutions. And that basically means evaluating the hyperparameters on a classification task and seeing what the results are. And then you would basically rank them via some selection strategy. In this case, I'm using tournament selection. And tournament selection basically is kind of like what it describes. You have a tournament size K, and K represents the number of individuals that will fight for selection. And so if you have a tournament size 4, for example, 4 randomly selected individuals from your population will be selected, and the individual with the best fitness, or in this case, the best classification accuracy, would become the parent. And so you perform tournament selection twice to get 2 parents, and then a crossover and mutation are performed like I described in the previous slide. And then the offspring becomes the parents of the next generation. So for a generation G, the offspring would become the parent for the generation G plus 1. And this is basically run until you have some chromosome which either meets your stopping criteria or you have reached your limit for the number of runs. And so in terms of experimental evaluation, genetic algorithms, because they're meta-heuristic and they're just one way of exploring a search space, they also have their own set of hyperparameters. And so the hyperparameters that are generally tuned the most in genetic algorithms are the population size, the mutation type, because you can randomly mutate individuals or you can follow some form of distribution. And the rate at which mutation happens is also controlled by something called mutation rate. And mutation rate is usually kept pretty low just because you don't want to change the amount of diversity that you introduce to the population too quickly. It should just happen at small rates, kind of like we see in biology where mutations happen at random increments. And then the crossover type basically determines how individuals will be recombinated, right? So if it's a single point, the 2 chromosomes are split up at 1 point and the genes are crossed over. And similarly, there's other types of crossovers that I'll be testing, but for the sake of the presentation, I didn't think it was worth going into too much. And again, there's a crossover rate which dictates the rate at which crossover is performed. And the crossover rate is generally much higher. And so finally, there's a selection strategy. I discussed tournament selection, but there's also different types of selections like rank and proportional, which can also be modified. And so in terms of progress, I've implemented the generic standard genetic algorithm infrastructure to test the experiment, and I've tested these on the traveling salesman problem with 40 cities and the point scattering problem, which is basically finding the number or finding the sum of the minimized distance of endpoints within an enclosed circle of unit radius 1. And then I've also implemented the baseline ResNet model just on the RATS data set, just so I have a baseline comparison for my results later on. And currently I'm integrating the encoding that I discussed with the standard genetic algorithm, because I now have the infrastructure via the first two problems. And now I'm also running experiments testing different hyperparameters for the genetic algorithm. And so what I have left to do is to improve the model performance by introducing data augmentations. I decided not to do that at the same time, just so that I wouldn't have too many confounding variables. And so once I have the hyperparameters in place, I'll introduce augmentations, and then obviously I have to compare the performance of the optimized hyperparameters to the baseline classifier to see if I was able to get any improved results. And as a stretch goal, SGA is just one type of optimization technique, but there are other types of techniques, of which I've listed a few, like PSO, TLBO, and differential evolution. So given the time, I will try to explore these to see if I can get comparable results. Some potential roadblocks for this problem is that the computational overhead is actually a bit complicated, because the number of functional evaluations in this case is equal to the number of runs, and for each run you have some number of generations, and for a given population size you need to run a number of epochs for each individual in the population, and for each epoch obviously there will be some evaluation. So the functional evaluations do blow up quite quickly, which is a little bit of a concern, because the quality of the population, or the quality of the solutions that the GA generates, is very much dependent on the population size. So if I'm going to be testing with large population sizes, then obviously the number of evaluations will grow quite fast. And so I'll have to see if larger models will be able to work with this with my compute resources. Otherwise, I may have to test on some smaller architecture. But I have yet to test that because I just finished the infrastructure for the GA itself. So that's something that I'll be testing next. These are my references.